Hello and welcome to the NIE Sustainability Learning Lab video series on water quality. Today, Grace Wee and I will be discussing methods to collect water quality samples as well as issues related to developing a water sampling program.
So Prof, this should be a short video. Why don't I just take the bucket over to the pond and collect the sample? Then we can move on to the other water quality videos.
We could do that, Grace, but we need to consider first what our objectives are in our sampling and research design. Let's start by considering the difference between a population and a sample. A population is the collection of all members of a particular group. So for example, the population of Singapore is all people living in Singapore. Now, if we wanted to know something about Singaporeans' preferences for food, we might go out and ask every single Singaporean what their favorite food is, and we would get our answer. The problem is that would be very difficult, it would be time-consuming and expensive. So instead, what we might do is go and ask a sample of 1,000 Singaporeans their opinion about food, and then hopefully we get the answer about the population. This means we're trying to draw an inference about the population from a sample. So a sample then is a subset of our population. It's easier to collect a sample, it's less time-consuming and more cost-effective, but what we hope then is that we can draw an inference about the population based on the sample that we collect.
I see, Prof. So if we think about the population of water in the Jurong Eco Garden ponds, this means that the population would refer to the water that's in the ponds right now, all the water that's been in there since the ponds were constructed, and all the water that will be in there until they disappear. That's a very long time and a lot of water.
Yes, great, so that's true. And if we wanted to know something, for example, about the dissolved oxygen levels in this pond, we would have to sample every single location, every second, every minute, every hour of every day for decades and decades. And that would be impossible. So what we hope to do then is by collecting samples in strategic locations within the pond, we can get some idea about the dissolved oxygen levels in the pond based on those samples collected. So Grace, let's get started. Our sample design and the methods that we're going to use to collect our samples will be dictated by a number of factors. These include the actual research question that we want to address, the time that we have in order to collect the samples, our budget and our resources available to collect samples, as well as logistical issues. And these logistical issues include things like how easy is it to get access to the water to collect a sample. Now, there are a number of different design methods that we can use to collect samples, so let's talk about some of these to begin with. One example of a design method is purposive sampling. With purposive sampling, what we're trying to do is identify sample locations with a specific purpose. In example here at the biotope, what we see is we might want to sample upstream of the biotope and downstream of the biotope to determine whether the biotope is having a positive impact on water quality. Or we might want to see if different land uses affect water quality. As we see in these graphs, for example, as we move downstream from peat forest to agriculture to urban land use, the E. coli levels increase. For the pond here at the Eco Garden, if we have a boat available, we might consider a random, a systematic, or a stratified random sampling scheme. Any sample location will be defined by a pair of X and Y coordinates, such as latitude and longitude. For a random sample, we randomly select an X coordinate and a Y coordinate, and that defines one sample point. You can use a random number generator to get the X and Y values. In this design, each point in the pond has equal chance of being selected, which is good because we do not want to bias our sampling, and therefore we may have a nice random distribution of sample points. However, with this design, as we see in this map, it also is possible that all sample locations are clustered in one area, which would not be representative of the entire pond. To avoid this problem of clustering, we might employ a grid and use a systematic aligned point sample scheme, as we see here. Alternatively, we may use a hybrid between full random and the systematic aligned point sample scheme. This is known as a stratified random sampling scheme, and each sample point within a grid cell is randomly selected. Finally, we might choose to take a transect approach to sampling. This method also frequently is used in biodiversity studies and beach surveys. The transect locations along which the samples will be collected may be determined purposefully or randomly in an area. Normally, with the transect approach, we would sample at regular intervals, and so this is a variation of systematic sampling. The transect approach is shown here for sampling done by NIE students in Lake Erie, USA. Water samples were collected along transects in front of a wild, natural beach, shown in the red pins, and in front of a groomed public beach, shown in the blue pins. Once we've determined our sample sites, we can identify the method that we will use to collect the samples. Generally, there are two types of sampling methodologies. There are manual grab samples, and then there is the continuous monitoring approach. We have the advantage of using both types of sampling here at Jurong Eco Garden. Grace, could you demonstrate for us, please?
Sure, Prof. Let's start by looking at manual grab sample collection. There are different instruments that can be employed for this type of sampling. The simplest is a homemade dipper, as we can see here. With the dipper, we can take our sample from the shoreline or from a bridge, and we will be collecting the surface water. Because you may not know where the dipper has been, it is best to rinse it 3 times before you collect the actual sample. An alternative to the dipper is a baler. The advantage of the baler is that we can cast it out further from the shore. The baler has a small stop ball at the bottom that will help to contain the water as it sinks. The baler will sink and collect the water. This means that the sample will be a mixture of water from different depths. As you draw the baler in, be careful not to drag it onto bottom sediment or bang into any obstructions. Once you retrieve your baler, insert the tip to release the stop ball and drain your sample. As with the dipper, remember to rinse your baler 3 times before you use it for the actual sample. Finally, we can use a Van Doren type sampler to collect manual grab samples. One advantage of the Van Doren sampler is that we are able to collect water from different depths of interest. This will allow us to compare how water quality varies with depth. To use the Van Doren, we first have to secure the plugs at both ends in the open position. Lower the Van Doren . Once it is at your depth of interest, release the weighted messenger down the line that will release the plugs at both ends. Once you raise the Van Doren, you can release your sample through the spigot. Remember to rinse your Van Doren sampler 3 times before you collect your actual sample.
In addition to the sample collection methods that Grace has just demonstrated, it's important to remember to record the sample location, the date, and the time of your sample collection. In addition, record any other pertinent observations such as the weather at that particular time. The location of your sample collection can be determined using the GPS capabilities on your smartphone and can be mapped out using a number of freely available apps for your smartphone. Also, remember to work cleanly. You don't want to contaminate your sample. So keep your fingers out of the samples, keep your fingers away from the spigots, for example, to avoid sample contamination. An alternative to the manual grab sampling that we've just observed is continuous monitoring with automated instrumentation, such as the YSI 6600, as we see here, that is located at the outlet of the Jurong Eco Garden. Grace, could you demonstrate for us, please?
Sure, Prof. This YSI has sensors that measure parameters such as pH, temperature, conductivity, dissolved oxygen, turbidity, chlorophyll A, and sample depth. Most of these parameters have been discussed in the other water quality videos. The chlorophyll A sensor really is a fluorometer that measures the amount of blue-green algae in the water. The parameters are measured every 15 minutes and the data is sent to a server via a 3G network. Once the data is processed, the results are available in real time on the Sustainability Learning Lab website.
The advantage of continuous monitoring is that it can place manual grab samples in a temporal context. For example, if we take a grab sample of water for dissolved oxygen at 8 a.m., we won't know what the dissolved oxygen is at 4 p.m. unless we stay on site the entire day or come back to collect a sample. From this graph of the YSI output, however, we can see how dissolved oxygen varies through the day or from day to day. So as you can see, Grace, there's a lot to developing a good sampling plan. Last, but most importantly, you need to work safely. This means when you come out to the field, you wear appropriate clothing, such as long pants, long sleeves, and a hat. Bring lots of water, as well as a safety kit. Also, never sample alone. Once you get on site, look for potential danger areas, like deep water or steep, slippery slopes. Remember, there's no sample that's so important to collect that it's worth risking your safety.